Hello students, so today we are going to discuss about one of the practical conducted in micro lab, that is under parasitology. So it is examination of malarial parasites. We can detect this parasite by different methods, but out here I'm going to discuss only about the smear method, which includes both thin and thick smear method. Okay, so this lab procedure, it plays an important role in the diagnosis of this parasitic infections, both for confirmation of clinical suspicion and for identifying unsuspected infections. Then the principle of this lab diagnosis are the same as in case of bacterial and viral infections, but the relative importance of the different methods varies greatly. Then, while isolation of the infecting agent and detection of specific antibodies are the major methods in bacteriology and virology, they are of much less importance in case of parasitology as well than the morphological identification of the parasite by microscopy. Okay. Then compared to the bacteria and viruses, these parasites are very large and possess distinctive shape and structure, which enables their specific diagnosis on morphological grounds due to their complex antigenic structure and extensive cross reactions. The serological diagnosis is of limited value in case of parasitic infections. Then, although many pathogenic parasites can be grown in the laboratory cultures, this method is not suitable for the routine diagnosis because of its relative insensitivity and the delay which it takes time. Then the morphological diagnosis of this parasite, it consists of two steps, that is detection and identification. Sorry, detection of the parasite or its parts in clinical samples and its identification. Sorry. Then the detection depends on collection of the appropriate samples and their examination by suitable techniques, whereas identification requires adequate skill and expertise in recognizing the parasite in its various stages and its differentiation from morphological similar artifacts. Okay, so what is the sample we preferred here is the blood sample. So this blood sample next to feces, it is the largest numbers of parasite are found in blood. The blood examination is the routine diagnostic method in this blood examination is the routine diagnostic method in malaria, filariasis, African trypanosomiasis. It is sometimes positive in case of Chagas disease and rarely in Kala Azar and toxoplasmosis. So we will discuss with the collection of blood, how we will collect the blood for detection of this malarial parasite. Okay, so for demonstration of malarial parasites, blood should be collected not during the peak of fever, but optimally several hours after the fever. Then bouts of fever follow the synchronous rupture of large number of parasitized erythrocytes, releasing the membrane shreds and contents. The emerging merozoids of parasites are the erythrocytes and initiate a fresh cycle of erythrocytic schizogony. The timing is particularly important in case of Plasmodium falciparum infections, as here the late stages of schizogony are not seen in the peripheral circulation or peripheral blood. Okay, so we are going to discuss how to prepare the smear of both thin and thick smear. Okay, so you can see here the slide. You can do both the preparation in one slide, or we can do it in separate slide as well. There is no issue, anyone is fine. So this is how we prepare a smear for microscopic examination or for microscopic analysis to detect the parasite. Okay, so a thin smear, how it is made, how it is prepared. So a smear is prepared from finger prick or infants from heel prick blood or ethylene diamine tetraacetic acid. Anticoagulated venous blood can also be used, provided blood films are made within 30 minutes. Then after that, a small drop is spread, around 10 to 15 microliter is spread around a clean, grease-free slide with a spreader to give a uniform smear, ideally a single cell thick smear. The margin of the smear should be well short of the sides of the slide, and the tail should end a little beyond the center of its length. Then this smear displays blood cells and parasites clearly as compared to the thick smear. Its only disadvantages is that only a small volume of blood can be surveyed, so that a light infection could be missed. Then if the smear are prepared from anticoagulated blood, which is more than an hour old, the morphology of both parasites and infected RBCs may not be typical. Then after drying the smear, it is stained with Gemsa or Leishman stain. For Gemsa stain, the smear is fixed in methanol for 3 to 5 minutes. After drying, Gemsa stain diluted 1 drop and in 1 ml of buffered water. pH 7 should be 7 to 7.2 is applied for 30 to 45 minutes. Then the slide is flushed gently with tap water. It is dried and examined under the oil immersion objective. Then after we examine, we will see that the cytoplasm of malarial parasite is stained blue and the chromatin dot is stained red. So these are the color we can see in the when we observe under the microscope. Then coming to the next stain, that is the Leishman stain. Here prior fixation is not necessary as the stain is an alcoholic solution which fixes as it stains. Then this Leishman stain is applied for 1 to 2 minutes and diluted with twice its volume of buffered water. The pH will be the same, 7 to 7.2, and is kept for 10 to 15 minutes. The smear is then dried and examined. So how we will report this thin blood film? In case of malignant tertian malaria, only the ring stage and gametocytes are seen in peripheral smear, while in benign tertian malaria, all stages of schizophrenia and gametocytes can be seen. Okay, so the this smear examination enables the appreciation of changes in the erythrocytes, such as enlargement, alteration of shape, fimbration, red cell stippling, which is also known as Schaffner's dot. This will be seen with Plasmodium vivax and irregular stippling, or we call it Morris clefts, as seen in matured Plasmodium falciparum trophozoites. Okay, so here the slide, it is shown both the thin smear and thick smear for the Plasmodium falciparum. This is a microscopic picture of both thin and thick smear. Okay, so any marked increase in white cell numbers and if indicated perform a differential white cell count. Then the parasitized erythrocytes are seen most often in the upper and lower margins of the tail of the smear. This is in case of the thin smear. Then we count the percentage of parasitized red cells when there is high falciparum malaria. Parasitemia will be 3 plus or more parasites seen in the thick film to monitor a patient response to treatment. And then a minimum of 100 fields should be examined before a negative report is given. Okay. So what are the parasites mostly found in the peripheral blood film? Which parasite? So in case of this protozoa, it will be Plasmodium. It will be Plasmodium species, Babesia species, Leishmania species, and Trypanosoma species. So these are the protozoa which can be found in the peripheral blood film. In case of helminths or nematodes, the parasites includes like Wuchereria bancrofti, Brugia species, and Loa loa and Mansonella ozardi. So these are the different parasites which can be found in the peripheral blood film. So here we have again the microscopic picture of Plasmodium vivax. You can see the ring form in both the thin blood film and thick blood film. Okay, so this is the chart of all the species under under the parasite Plasmodium. We have different species, and at the same time the stages of all this Plasmodium is given here. So we have the ring stage, the trophozoite stage, schizon, and gametocyte. So these are all the different stages of the Plasmodium. Okay, so with this we finish with this topic. Thank you.